lay it out. So we'll talk about first talk about retroviruses, and then talk about the Virology. There's only one example of the gapped a DNA virus that actually requires reverse transcriptase. So both retroviruses and the DNA virus require reverse transcriptase. They're not obviously in the same classification or family at all. But the reason why I group them is because they both use reverse transcriptase, right? And I want you to see the strategy of using reverse transcriptase. For the retrovirus, I'm going to actually talk about two different viruses, and there's a reason why. The first one today, I'm going to be not hand-waving, but give a general, not at all in-depth in terms of attachment entry at all about Rao-sarcoma virus, which is a retrovirus, and then starting next week, I will talk about the HIV, human immunodeficiency virus. The reason why I'm looking at both is that for today, since we just came off from talking about the polyomavirus, which is a tumor virus, so I'm going to continue, so that's my bridge, is continue to, since I mentioned polyomavirus, I said that it can be a tumor virus, but I didn't say anything about how tumors are formed, and this is not a cancer biology class, but I do want to give you a sense of, there are a lot of tumor viruses. A lot of people study probably tumor viruses and any other type of viruses because we do want to treat the disease. So I'll give you a kind of a more of a laboratory application to understanding viruses, right? What is a tumor virus? How does that cause cancer, and so on and so forth? So that's why Rao-sarcoma virus. Also, it just happens that my doctoral work was on Rao-sarcoma virus, so it's just easier for me to talk about it than just about anything else. So we'll just spend some time today and then talk about HIV, and HIV is a big, huge area. It takes a long time to cover, so it might take more than 2 lectures for me to go through HIV, and then hepatovirology, and then the last portion would be 1 lecture on your immune system. How does your immune system address a viral infection? In immunology, we talk mainly about bacterial infection. In this class, the reason why I do that is that I know that many of you who go into, most people who take immunology and virology go into CLS, and so I don't want to be too redundant, right? Or, yeah, too redundant. So that's why that's going to be the last part of this. And then the 4th exam, okay? So that's the setup lecture. So retroviruses. This is the family, right? Family of retroviruses, and under the classification of RNA virus, that requires a reverse transcriptase. Okay, most, at least in the book, but I can't think of a retrovirus that is not also considered to be a tumor virus. Well, actually there are. Take it back, there are, but a lot, or most retroviruses are also tumor viruses. Okay. So that's that. There are for Rao-sarcoma virus, it's in the alpha Virology virus. This is the genus. This is a genera. Okay, that's Rao-sarcoma virus. Rao-sarcoma virus. So retrovirus is a family, and basically that falls under the classification of RNA virus plus reverse transcriptase. There's alpha, beta, gamma, and epsilon. There are a whole bunch of genera, and each one has a specific species, strain of species. Lentivirus is another genera. That's where, when we talk about HIV, so HIV, human immunodeficiency virus, it actually falls under the lentivirus. Okay. they use very similar system. So the Rhinosarcoma virus uses a similar system for virion replication as HIV. So again, that's why I'm going to reserve talking about the specific mechanism of infection for HIV and for Rhinosarcoma virus. I just want to introduce basically what it is, what do we know about it, host range, and then leading to why is this a tumor virus? What does it have to do with a tumor virus? When we talk about HIV, we'll talk about how people with HIV, with not just HIV positive, with AIDS, right, has a higher chance of developing cancer, which is slightly different from how Rhinosarcoma virus very specifically, and this is one of the few agents that we can say specifically causes cancer, not just increases a chance, but 100% of the time we see that it actually causes cancer. Still, it still increases the risk factor, but the risk factor is so high that it's practically 100%. Okay? You buy that explanation? Okay. Then we're going to talk about Rhinosarcoma virus first. Okay. Again, minus all of the mechanisms so far. The reason why it's called Rouse is because somebody named Dr. Rouse studied it, so that's why it's Rouse. Sarcoma is because this virus infects mesenchymal cells, mainly fibroblasts. Okay, and in cancer, cancers are divided into two major, and there are a few others, but two major identity. One is called sarcoma, and the other is carcinoma. Carcinoma, there is more prevalent. Carcinomas originated from epithelial cells, basically, okay, or epithelial origin. Whereas sarcoma originate from mesenchymal cells or origin. Okay, so for instance, fibroblasts, you can have cancers that lipoma or liposarcoma is a cancer that came from fat cells. Chondrosarcoma is a cancer that came from chondrocytes, which are cells that will produce the cartilage and so on. Osteosarcoma, so mesenchymal cells. Okay, so those are sarcomas. Again, like I said, most Rhinosarcoma virus or RSV infects mainly the fibroblasts, and the host range varies. Very specific host range, chicken. So I don't recommend you do this, but you can drink a test tube that has tons of Rhinosarcoma virus, and it shouldn't affect you. Okay? Shouldn't, okay, but don't. Shouldn't. Nobody has ever done it, so we don't know. But you shouldn't do it anyway. Okay. The infectivity of Rhinosarcoma is extremely high. The MOI, meaning the multiplicity of infection is 1. Meaning that if you have one virus that can infect one fibroblast cell. So in the laboratory, it's very easy. You just take some of your cells, dump any amount, literally any amount of virus into the culture medium, and you will produce more, more cells. Okay, and these cells, the viruses don't actually induce tissue damage. And so here's some explanation. So remember for polyomavirus that when the polyomavirus actually does induce in permissive cells, right? If you remember, tissue damage is incurred. But when the polyomavirus actually induce or increase the chance of cancer formation, remember it's in non-permissive cells. right? So if a virus wants the cell to become cancerous, it doesn't want to destroy the cell. So therefore, no tissue damage, right? Same with here. Same with any of these retroviruses. They don't usually, not that they don't do it, but they don't usually induce tissue damage, because most of them, again, or they will go into the latency phase. Latent phase, not latency phase, latent phase. Okay, that's when they cause damage right here. Okay, so they do have a lytic phase, but in the lytic phase, again, the cancer forms when these viruses infect the cell and then goes into the latent phase. Okay, parallel, very similar to polio virus. Okay. All right. So the MOI, multiplicity of infection, is 1 to 1. That's sarcoma. For all of the retroviruses, they're RNA viruses, and they contain quite a few genes. Okay, not quite a few. They contain a few genes. Okay, and all of them are divided to So what happened is that they each also have, remember the transcriptional units. So they have 3 transcriptional units. Okay, again, we're not talking about polyprotein. We're talking about transcriptional units. So that these 3 transcriptional units are referred to as GAG, POL, and ENV. GAG, POL, and ENV, and then each of these transcriptional units, when transcription is carried out, then alternative RNA splicing would yield different mature mRNAs, leading to the production of different proteins, right? So that's basically the same. So the GAG basically encode for capsid proteins, capsomeres. The POL encodes for basically reverse transcription and anything that has to do with the viral genome synthesis or replication. And N, because retroviruses are enveloped viruses, so the envelope encodes for envelope protein. Okay? Yes, ma'am.
This is all retroviruses?
Yeah, all retroviruses do this. We'll talk about sarcoma specifically because each of these genera of viruses have a slightly different last part, the 4th part. And sarcoma virus just happened to be this, and then again, other retroviruses might have other special genes, okay? Yes, ma'am?
Is it positive strain RNA or negative?
It's plus strain, and they actually, and again, all of these mechanisms I'll talk about when we talk about the lentivirus. There's basically plus, but 2 molecules of the plus, and there's a reason why that is also. So it's not double-stranded, but there are 2 molecules of both of them are plus. So for sarcoma virus, there's a gene called the SARC gene. It's actually SARC as in this, but then people abbreviated this. SARC stands for sarcoma, of course, but it's a SARC gene. Okay, that then will encode for the SARC protein. The SARC gene is referred to as an oncogene, which is a slight misnomer, but that's okay. And a SARC oncogene and SARC protein is referred to as an oncoprotein. An oncogene, oncoproteins have to do with the fact that they're called that because they either greatly increase or they initiate or somehow is connected to the formation of a cancer cell or pushing the cell to become cancerous. Okay, so the bulk of the explanation is here. Okay, but then the idea of what we know as an oncogene. started with the study of sarcoma virus as well. So this is more, some of this is a history lesson as well to give you an idea of how powerful viral infection is. And we talk about things like, you know, viruses evolve and so on and so forth. It's all embedded in this, okay? So far so good. So cancer, what does that mean? That means that for the most part, there are three characteristics of most cancer cells as well as tissue. One is that they have uncontrolled cell proliferation. That's one. Okay. So these cells divide even when your body doesn't want them to divide. And again, we're really talking about for sarcoma virus, the chicken, right? So chicken do have practically all organisms that we know of have tumors, form tumors, even plants. Okay. Uncontrolled cell proliferation. Most cells, practically all cells, all normal cells require something called contact inhibition as well as anchorage dependent growth. But cancer cells don't normally have that. So cancer cells don't grow through contact inhibition, don't go through anchorage dependence. I'll explain what all of these means. And then three is that for the most part, there are exceptions, but for the most part, cancer cells don't undergo apoptosis to die. Basically, they don't die. They do die though through the process of, so no apoptosis, instead they go through a process of necrosis. That means that they run out of nutrients, run out of oxygen, and they just kind of waste away. Right? So that's by necrosis, but no apoptosis. So the idea is this. So the idea is, let's look at this. This is important. Most of the cells in your body, including blood cells, right, including hematopoietic stem cells, when they divide, two things they have to consider outside of the signal to undergo cell proliferation. One is that even blood cells that normally swim in the plasma have to be anchored somehow to either the extracellular matrix or interacting with usually a cell, right? Like the stromal cells in the lining of the bone marrow, for instance. Okay, so they have to anchor. So that's anchorage dependent. Cancer cells don't have to do that. They can divide in a solution. Contact inhibition is, and this is important. This is why you have an outgrowth of tumor. Is that most cells, they only divide if there's space for them to divide. So if an organ is this big, right? Then if there's a little bit of space, then the cells will divide to fill in that space. But once that space is occupied, the organ will not get bigger in any way. Okay? And they know that by contact inhibition, so that a cell that is nudging against either another cell, usually another cell, or an extracellular matrix, it will not divide. It will be told to stop dividing in that direction. Okay? Cancer cells don't do that. That's why they have outgrowth, right? So if you have an organ that way, and then you have a bunch of cells already in that direction, but if this is a cancer cell, what happened is that it continued to divide, and now you have an outgrowth, right? Right. So that's what happens there. And then on top of that, then you have the number one, which is all of your cells, normal cells in your body or non-cancerous cell in your body, if they need to divide, they have to be told to divide by a number of different signals, right? In this case, cancer cells don't require the typical signals. Sometimes they send out their own signals, and sometimes they have this positive feedback loop that they will continuously go through cell cycle again and again and again, as long as there's enough nutrients. So that's that. And then again, they don't for the most part, they don't die. So by apoptosis, so even if there's an outgrowth, they're not told to stop dividing. They're not told to die so that you can reshape the organ, right? So that's that. Okay. The Src protein basically targets that. Src protein causes a cell to undergo uncontrolled cell proliferation. Usually, in most organisms, it takes a number of different mutations on a number as in like half a dozen really important genes that normally have to be expressed to regulate the cell control. Normally, it takes a lot of mutations. That's why in the formation of cancer, it takes a long time to form. Cancer takes a long time to form, decades. But the Src protein can control all of those just by the Src protein alone. Right? I think in counting, there are probably 50 plus substrates for Src. And they all have to do with cell proliferation and cell differentiation. Okay, so that one protein is important. Where did that oncogene come from? Where did that Src protein that is found in the Rhabdosarcoma virus? Where did it come from? Okay, is the next thing. And for many, maybe most of the tumor viruses, this is how it happens. Okay, so are we good with cancer? And so Src addresses this. Okay, so now let's bring back. Let's erase all of this, and let's talk about genetics. So sorry, we have to talk about genetics. Usually genetics is easy, right? Most people like genetics. I got hooked on cell molecular because of genetics, so I understand. So a virus, let's say here's a viral genome, right? Contains a number of genes, and here's the Src gene right there. Right? So that's the Src gene right there. Okay. We call that Src gene. We call that either the gene or the protein is still Src, and we call that V-Src. V stands for viral. Yes, sir.
In like testing situations, would you be okay if we wrote Src as S-A-R-C? Because you wrote it originally?
You can, but this is when you read papers, this is really the way that they write it.
Okay.
But any way you want to, it's about the same. Okay. In your own cells, chromosome, you also have a Src gene. We call that C-Src. Stands for cellular Src. So the virus contains the same gene that we do. Why is that? Which one came first? I can tell you that it's our C-Src came first. Okay? So that's why the C-Src is referred to as the proto-oncogene. So people actually discovered Src first, and then they thought, oh, this is a viral gene, and let's see what it does, and lo and behold, we have it too. But they're different. Okay? So in fact, this gene right here, the V-Src, is shorter than the C-Src. Okay? So again, this happens again when you have the viral oncogenes. This is probably the way that happens. Of course, we couldn't turn the clock back and see if this is actually it, but the indication is that this is how viruses have acquired these oncogenes. And C-Src itself is not an oncogene. It's a proto-oncogene. It's before the oncogene. So the oncogene is really V-Src. C-Src is not called an oncogene. It's a normal gene that performs a normal function. What is a normal function? Cell proliferation. Regulated, controlled, regulated cell proliferation. It's when something goes wrong with that, which is in the form of V-Src, okay, that's when. So carcinogenesis might occur. So there are like a number, just 2 or 3 different big ideas that come from people who study Sark. Okay, and I'll point them out, including phosphorylation, right? So being a kinase. So there are, like I said, a number of big ideas that we now see prevalent in a lot of different systems. Okay, just from studying Sark, the Sark protein. Okay, so far so good. So now, how does the sarcoma virus, the virus, acquire this gene? Well, we have to understand roughly how infection occurs, right? So a virus, let's say, who knows how long ago, hundreds of thousands of years ago, I don't know. You have a virus that contains RNA virus that contains the GAG, PAL, and gene transcription units, but no Sark gene. A long time ago, right? That's what they do, okay? And what happened is that after infection, all retroviruses do the same thing. They convert the RNA into double-stranded DNA. Okay? And if you have a cell chromosome like this, the viral double-stranded DNA, in order to prevent the viral RNA, double-stranded DNA from being digested, because it's just a small piece of DNA, and remember, DNAs would like to digest it, it's not circularized. So what happened is that and I'll give names to all of these things later, provirus and all of that, but right now just know that double-stranded DNA is going to get inserted into a host, and basically now you have, right, so this represents that viral DNA inserted into the host fairly randomly, but not completely randomly, right? There are certain insertion sites, but fairly randomly. So far, so good? Now, okay, for viral replication, what is required? It requires that you have to have viral proteins as well as the viral genome. What is a viral genome? It's an RNA viral genome, so transcription occurs, right? Transcription will occur normally through this viral genome, and that's how you get many more viral genome during nucleic acid, and then get packaged, and then get sent out, right? Transcription, if you remember, how do you terminate transcription? Is there a transcription termination site? There isn't, right? Polyadenylation is used to terminate transcription, but it is not a transcription termination site. So therefore, there isn't really a site right here, right? You have polyadenylation site. Are you following me so far? Now, how effective is a polyadenylation site? Does it always terminate transcription? No. That's why you actually have multiple polyadenylation sites, right? Usually it's for either, multiple polyadenylation sites have a couple of roles, function. One is to maybe you might want to transcribe a gene, but you don't want all of the gene to be transcribed, so the last few domains might get removed, and then you have slightly different versions, alternative versions of proteins, right? So that adds to diversity of the number of proteins that the cell actually expresses without having to have multiple genes. Okay? You kind of remember that, right? But another reason to have multiple polyadenylation sites is just in case one doesn't work, the next one can stop, right? And then if the second one doesn't work, the third one, usually that's enough. Okay? So far, so good? All right. So let's say that this right here, this viral DNA, were inserted where it's very close to a C-Sark gene. let's say the CSAR gene is right here, it might clip a little bit of the upstream coding region, but very close to here, so that during viral DNA or viral transcription RNA synthesis, right? The polyadenylation site isn't sufficient. There is only one. So what will happen? What will happen is that transcription continues. Where on earth do you find another polyadenylation site all the way toward the downstream from that CSARC gene? Right? That CSARC gene also has multiple polyadenylation sites. Let's say it does that and it only synthesizes a portion of the CSARC gene. Now the new viral genome not only contains all of the GAG, POL, and N transcription unit, but also contains the CSARC gene, but not the entire thing, right? Only a portion of it. And the short version of the take-home message is that piece of the CSARC gene is really important in controlling cell proliferation. Without it, you get uncontrolled cell proliferation. Right? That's the take-home message is that. And we'll talk about what that encodes for. Okay. But so far so good. So most of the oncogene, viral oncogenes we're guessing were acquired through this way, and not just RNA tumor viruses, but also DNA tumor viruses, because DNA tumor viruses, some of them can also get inserted into the host, right? But instead of replicating, to undergo transcription to replicate the viral genome, you undergo DNA replication to replicate the viral genome. Yes, ma'am.
Could you just repeat what you said about polyadenylation site? Because I don't really understand.
What do you mean? Which part of polyadenylation site that I need to repeat?
Okay, when you talk about the viral DNA being inserted, I don't really get what happened.
You know what polyadenylation site is? Right. So that's a site that allows a cell to add the poly A tail to the transcript, but there are multiple polyadenylation sites. And polyadenylation then would cause the cell to terminate transcription, right? But there are more than one, usually, not always, but usually more than one polyadenylation site. So if one polyadenylation site, let's say the viral genome has a polyadenylation site, but and the reason for that polyadenylation site is because, the reason for having multiple polyadenylation sites is because if one is not sufficient to terminate transcription, then maybe the next one will do it, right? So you don't want transcription to transcribe the entire chromosome, right? You want to terminate at some point. So during viral genome replication, right, maybe